discrete methods. So the session this morning is discrete methods. For the first speaker, Rudy, I think the discrete part of this is probably more that the underlying algebra of the tropical geometry. Perhaps maybe she'd have something to say about that. But I think, Rudy, it might be nice just if you start with a little bit just about exactly where you are, because you've said in your talk you just got there the Naval Graduate School, but maybe you could just talk about where physically you are right now, just as you start the talk. That'd be great. Take it away.
Thank you so much for having me. And yes, I'm at the Naval Postgraduate School in California right now. And yeah, so last moment, I changed my mind. I thought I was gonna talk about tropical support vector machines, but I would like to solve some open problems on relating to tropical PCA. So I'm going to talk about this one. So here we go. All right, so this is, okay, of course, like everyone knows phylogenetic trees, so I'm not going to talk about the details, but this picture, so this is a data set kind of motivated me to think about PCA over the tree space. So this is the data set relating to African coelacanths and lungfish. So you see this that like some, okay, so if I can draw some pen, and I'm sorry, I'm still kind of new to this one. So this is the lungfish. You see this lungfish and the coelacanths, and also here you see this. Those are tetrapod that's relating to like, mammals and so on. So that's the living things in the land. So basically the original data set, that's a whole sequence on African coelacanths that's published in Nature, like that's 2013. And immediately after that, people published a paper on all like sequences on this genome. So that's in molecular biology and evolution, that's 2013. So I use this data set and see like, how the gene trees are distributed over the tree space. That was the motivation. And also the biologists are really interested in this data set because they ask the question, this lungfish or coelacanths, which one is closer to mammals or like tetrapod? So that's actually relating to the like really classic question from Biology 101. That's how we migrated from ocean to land. So that's the really big question they try to answer. But you notice the phylogenetic tree they reconstructed, it's not well resolved. So they couldn't really figure out the lungfish or coelacanths, which one is closer to tetrapod. Okay, so my interest is, as I said, I'm interested in phylogenomics than phylogenetics because I would like to do some statistical analysis over the set of phylogenetic trees instead of just reconstructing one phylogenetic tree. So I would like to see how phylogenetic trees are distributed. So basically what's going on here is that in general, we have a bunch of sequences for each gene, and then like reconstructing phylogenetic gene trees, and then we vectorize somehow, and then just we're gonna have a set of vectors. So I'll talk about, oops, that's annotating thing is something there. And I try to erase, I'm sorry, I didn't realize. All right, so yeah, so I'm gonna tell you how we vectorize and so on. And in this talk, I assume that we got a good set of gene trees. So I assume that gene trees are well fitted. Okay, so this is a big assumption, yes. But in order to solve this problem, I have to start from somewhere. Okay. All right, so this is in a paper I wrote with Fukumizu-san and Chrisa Face, and that's 2017. And just we kind of, try to apply just classical PCA. We applied a bunch of like clustering method on a Coelacant data set. And we saw that this is kind of hard to see because it's kind of three-dimensional PCA, and it basically we saw this two clear separation. And I thought, oh my gosh, this is awesome because, the reason why this phylogenetics, we couldn't really resolve because I think we had two big clusters. But I looked at the picture really carefully and it seems like, somehow that it looks like tree space. That's kind of, I felt really bad feeling about that. So this is a motivation why I wanted to, actually Tom too, I worked with Tom for developing PCA with BHB metric. But also this is really, really beginning that I wanted to actually see this like some PCA over the tree space. All right, so here, just I'm pretty sure many people know the PCA, and PCA is a great tool to visualize the data set, how data points are distributed over the space. So basically, it computes the principal component, first principal component, second principal component, and so on, and reduce the dimension. That's what they projecting it, and onto the actually lower linear space, and then try to visualize. So here, the problem is that we have a set of gene trees. Somehow I vectorize it, and then I would like to visualize how data points are distributed over the tree space. Okay. All right. So there are so many ways to calculate the PCA, but here I would like to just define this way because the tree space is, first of all, it's not really Euclidean space. So that we cannot just directly use classical PCA. So somehow, and also that it's not Euclidean space. So you have one definition of the PCA doesn't really coincide with another definition of PCA. So here I define the PCA as a like kind of linear space such that, so this blue points are data points, and the red points are projected points onto linear space. And then basically we take the sum of the distances between red point and blue point. And then we try to find the linear space such that the sum of the differences between red point and blue point is minimized. So this is, we define PCA as a linear space, such as minimizing sum of the residuals. All right. Okay, so for tropical metric, it's natural to use the equidistance tree. So equidistance tree is a phylogenetic tree, that's a rooted phylogenetic tree, such that root to each leaf has the same distance. So for example, this tree, so we have root and leaf A, that's a distance 2, root to leaf B, that's 1 plus 1, that's equal to 2, and so on. So it has all same height. So I assume that all gene trees are equidistant trees. Okay, so this is a big assumption. And actually, one way I'm gonna say this assumption is not too bad, because if you think about the coalescent model, that's basically the same assumption. So I kind of assume that we have a multi-species coalescent model. All right, so then I'm gonna vectorize each tree. So first, I'm gonna calculate pairwise distances. So this is the dissimilarity map or distance metric. And if you calculate, so if we have, so again here, I assume that we have perfect set of gene trees. And from each gene tree, I calculate the distance metric, and then that's automatically that's a tree metric. And so we have a like diagonal equal to 0, and we have the ij's entry of this metric is a distance from a leaf I to leaf j. Right. So for example, if I pick the AB code like element here, that we have 4, and from leaf A to leaf B, that's a 2 plus 1 plus 1, that's equal to 4. And for example, BC coordinate, that's a 2, and we have B to C, so 1 plus 0.2 plus 0.8, that's equal to 2. And that's a diagonal, diagonals are all zeros, and also that's a symmetric. So I take the just upper triangular part of the metric, and I order the element lexicographically, and it becomes vector. And you notice that since this is an equidistance tree, we 3-point condition satisfied, so it's 1-to-1 correspondence. So I define the tree space as a space of ultra matrix. So this is the space I'm dealing with. Okay. I hope no questions, or if you have a question, just let me know. So far, I think so good. So now I'm gonna talk about tropical algebra. So first time I deal with tropical algebra, I was so scared. I saw this when I was in Berkeley, and that was the scariest thing ever. This part is easy. Okay, so we deal with max plus algebra. So A and B are real numbers. And so A plus B, I define as max. And A multiplied by B, that's addition. That's a classical addition. That part is really intuitive. But beyond that, it goes like really weird to me. But now it's my friend. I really like this space. So, okay. Then, so we have to define the distance, right? We are dealing with space of ultra matrix. So we have 2 ultra matrix. Then I have to define the distance between them. So this is a tropical distance, and it's also called the generalized Hilbert projective metric. All right, so V and W, those are ultra matrix. And in a sense that you can also think as equidistance trees, okay? Because that's a 1-to-1 correspondence. And then basically that's this crazy definition. But all right, so I'm gonna draw the notation. I like actually drawing things, and so this is kind of hard for me. But also you can think that that's the max of V minus W, and this is equal to, that's also, oh, sorry, that's a minus. So sorry. So that's a minus mean of V minus W. That's also, you can see, this is exactly same. Okay? So here, E is the basically N choose 2. All right, so that's the basically dimension of the ultra matrix. All right, so basically in a sense, so intuitively the tree, that's kind of also, this is again, I'm gonna talk, that's connected to the open problems, and I'll talk about that. But basically, you can think that the biggest difference of the pairwise differences between the leaves in one tree to other, and minus the smallest difference between 2 like pairwise distances. So that you can kind of think that way. So also another thing is that I've been working on a tropical SVM, support vector machine, and I realized that it's also very robust against outliers or noises. And also that if you increase the dimension, it's pretty not, like it's really not sensitive. So this is just FYI. So this is one good point. All right. Some reason it's a stop. All right. So, gosh, I'm sorry. All right. So this is example that like V equal to 0, 0, 0 and W equal to 0, 3, 1. Then you take the difference. So you can take W minus V. It doesn't matter which one is first. And then you're going to get like max of the difference is 3 minus mean of the difference that's equal to 0. So 3 minus 0 equal to 3. So that's the distance. Okay. So that's the example. All right. So annotating. Yeah, this is going to be really interesting. All right. Okay. So now, oops. All right. So now, so in a first paper, we wrote the tropical PCA. We defined the tropical PCA as a two things that's basically tropical linear subspace and also tropical polytope. But the tropical polytope is much more suitable for space of ultramatrix. So we focus on the tropical polytope. And I will talk about that, like why this is more making sense. All right. So tropical polytope, that's or tropical convex hull. That's a convex hull of tropical convex hull of finitely many vertices, and just defined as this guy. All right. So actually, I want you to think about this. So in a real, like classical Euclidean space, if we have the like all vectors, V1 through VR, and A1 through ARs are actually some real numbers, and instead of tropical multiplication and tropical addition, these definitions actually define the linear space, right? That's the just classical linear space. So this is the intuitive why we actually use the tropical polytope. And also the tropical polytope is a tropically convex. So that it has a nice propositions, a lot of nice propositions. This is why we decided to use the tropical polytope as a, like, to compute the tropical PCA instead of tropical linear space. Okay. All right. So, and also another thing is that this is kind of not intuitive, but we actually use like all tropical arithmetic over the tropical projective space. So that's basically modeled by 1, 1, 1, 1, 1. There is a reason. So I'm gonna go back. So if we don't deal with that over the like tropical projective space, modeled by 1, 1, 1, 1, 1, 1, or constant, this metric is not anymore metric. So in order to make this tropical distance as a metric, I have to model by all 1, 1, 1, 1, 1, 1, those vectors. Okay, so this is just a note. And so this is another example. All right, so we have 3 points. So that's the red points are that's the vertices generating the convex hull, 0, 0, 0, 0, 3, 1. So those are, all vectors. And I take the tropical convex hull of these 3 vertices, and this is a tropical polytope. This just by coincidence, it's actually, this is equal to classical polytope, this case, but it doesn't have to be. I will show you more example in a later. Okay, and also another tool we need is remember, So here, actually, I'm gonna go back. I'm sorry, go back and forth. All right. So I defined that, so I wanna find, so now problem is I wanna find the tropical polytope such that minimizing some of the distance, tropical distance between red point and blue point. Okay? So that's the problem. So now this is the official problem. So we have the tropical polytope. We define that. And another, so, and also we have tropical metric, tropical distance. I have that. So another tool I need is projection because I have to calculate those red points. I have a great formula. That's all thanks to Barn. He told me a bunch of stuff. All right. So this is the, so oops, I went too back. So this is a projection. So suppose I have the tropical polytope, then I want to project another point that's a D. So D1 through DS, those are vertices of the polytope. And then just I want to project this D onto this polytope. This is just a formula. So there is a concrete formula. So this is nice. And also the another nice thing is that this is actually a problem we had when I was writing with Tom and Grady on PCA or with BHB metric. Basically, convex hull in terms of BHB metric, its dimension was crazy. Just dimension goes up like basically just unbounded. It's just crazy. So, but this case, if you think with tropical metric, then actually dimension of the tropical convex hull is like acting as exactly Euclidean space. So suppose I have the tropical polytope with S many vertices, then the dimension of the tropical polytope has at the most dimension S minus 1. So this is nice. This is why we are using this tropical polytope for the computing tropical PC. Okay. All right, so this is the projection. So I want to project this D onto this polytope. I just plug in the formula. I just show you. And this is just, it becomes 035. So that's nice.
Ready?
Okay.
Can you hear me? Ready? Ready?
Oh.
Hello?
Yes.
Just 8 minutes.
Huh? 5 minutes. Yeah, it's almost done. So this is okay. So 5 minutes. Yes, I will try to finish off. All right. So this is actually
8.
Huh?
8 minutes.
5 minutes. Yes, I will do that. It's almost done. It's almost. I promise. So this theorem, I will talk about this a little bit with example because that makes sense. So I'm gonna skip this. This is actually Coelacant's data. The problem is about 1/4, 1/4, 1/4, 1/4. Gene trees have those tree topologies. That was the problem. So this is a tropical PCA I projected onto tropical triangle. And those are tree topologies. And I color coded because one big question we have, still it's open problem. I really want to talk about. You notice that you have a bunch of cells. And black dots are actually, that's kind of, tail distribution. So you can ignore this. But you notice that in a cell, they have the basically same colors. And this theorem we showed is that basically, if you take 2 points from the same cell, they have to have the same tree topology. That's kind of we showed. So now question is that's the biggest question. For the BHB space, we know exactly the tree topology looks like, right? How they change tree topology over if you cross the orthan. But for these cells, they have the same, within a cell, I know that all points have the same tree topology. But if you cross it, I have no idea how they change the tree topologies. So this is the biggest question. And more, concretely, if we have a tree like tropical polytope computed from the ultra matrix, and we know the ultra matrix and equidistance tree, that's a one-to-one correspondence. So I have no idea how I can combinatorically explain the tropical polytope in terms of tree topologies. More precisely, one more level down, if I calculate the tropical line segment, that's the basically tropical polytopes between 2 points along, that's actually tropical geodesic, and how tree topologies are changing along the tropical line segment, tropical geodesic. I have no idea. So this is actually big open problem. And if you are interested in, just let me know. And so this is a P complex, another example. And you notice that also same kind of tree topologies. And I actually apply to the influenza sequences. And I compared with BHB, PCA, and we got a much better or much higher R squared. In this case, it's not R squared, but it's R. But we have the better, higher score. So anyway, but this is published and it's in the bioinformatics. And if you have a question, let me know. Thank you so much for your attention.
Thanks. So where am I? Thanks. Thanks very much, Rudy. Great talk. Great to kick off Phylomania with tropical geometry and tropical algebra. There you go. Bang. Here we go. Okay. So I should have introduced myself at the start as well. By the way, I'm Jeremy Sumner. For those who don't know me, but does anyone have any questions for Rudy? You can put questions in the chat, or you can. I don't think people can jump in now. I mean, maybe put up your hand first so we get a feeling for how many people are going to ask questions. But does anyone have a question for Rudy?
Sorry, maybe I actually just talked too fast.
Oh, it's okay, because it's my job as chair to ask you a question. I'm just giving people time. No, okay, so my question. Oh, yeah, you go.
I just have a quick question. You said that they don't, your polytope doesn't always, your convex hull doesn't always look like a polytope.
Yeah. So
could you show us something that doesn't look like one?
Yeah, so just a moment. Sorry, oops, this is the wrong way. So this is actually, for example, this is a tropical triangle. So you notice that just one bird, it is a tropical triangle. And so like we know the tropical triangle and so on, but the problem is that so tropical geometry doesn't care about tree topologies. So interpolativity, so now we have trouble because I can calculate tropical triangle or square or whatever, but the problem is that I just don't know how it's changing in terms of tree topologies. And I don't know, like I cannot connect to combinatorics in terms of trees. And this is actually kind of, I really want someone to solve this problem because it's not so bad to compute the tropical triangle. It's very fast, but just I think the biggest obstacle I have so far is the interpolativity because BHB is so nice and popular because you can interpolate in terms of trees. But yeah, so I just cannot interpolate, and this is my hardness.
Okay. Thanks, Rudy. I'm going to cut. There is a question here from Jonas, but I think what we'll do, we'll just keep moving. And what we'll say is, when you put your questions into the chat, we'll use those as well. But afterwards, because the talks are being recorded, the chat is also recorded, so the speaker will be able to access those questions. So you can ask questions to the speaker in the chat, and we'll try to make sure those questions get to the speaker as well. But I'll just move it along. So thanks very much, Rudy. Okay, so next is Michael Hendrickson, who assures me he's ready to go. So are you there, Michael?
Sure am. Can you see me?
Yep, hello, Michael. You've got a share screen or something, or how are you doing the
sure
talk?
do. Yeah,
Yep.
Share screen.
So Michael's in Henrik Heine University. He's been there for this fantastic year of 2020, I think. Must have moved there shortly before, unless I've got my dates out of whack here. But anyway, again, so in the spirit of just give us a quick shout out to where you are and maybe what time it is and what the weather's like outside. And take it away.
All right. No worries. Hello, everyone. I'm Michael. It's 1:45 here at the moment. A bit chilly. I don't know if you can see. I'm wearing a jacket at the same time. I'm in Dusseldorf, which has been very nice. I did arrive just before COVID hit, but it hasn't been all bad because there are many squirrels here, and there's a hedgehog that lives just outside my apartment. So very exciting for me that way as well. So I guess I'll get into the talk. Just confirming everyone can hear me properly.
Yeah,
Yep.
coming through loud and clear.
Excellent. I just know my microphone is a little bit finicky, so if I cut out at any time, just yell at me, please. Anyway, so as is mentioned, I have moved to Germany for this year to live amongst the biologists and observe their ways. I'm going to be talking today about incompatibility and universal tree sets. And this is a topic that actually came out of a discussion with one of the biologists who live here. They were asking me if they have two splits tree networks with different numbers of leaves, how can we compare how incompatible they are? Like which one has more discordance? And I thought about that for a little while and came up with the following definition of compatibility, which is different to K-compatibility, which some of you may be familiar with, but I'll get to that in a second. So this definition, we say that a split system or a set of splits has a tree set of size K if you can find a set of K phylogenetic trees that display every single one of the splits. So for instance, here we have the set of splits listed here, 1, 2 versus 3, 4, 5, and so on. You can see 1, 2 versus 3, 4, 5 corresponds to this edge here. 2, 3, 1, 4, 5 corresponding to this edge here, and so on. And we can see, in fact, that this is a minimal tree set, which follows quite naturally because we have 5 splits in our set here, and each tree can only have at most 2.
I don't have to wear my mask because I've been vaccinated.
Sorry, Scott.
Well, exactly, but
Sorry about that, Michael. Keep going.
No worries. Sorry, yes, I was saying this set of splits has 5 splits in it, and each 5-leaf tree can only display 2 splits, so you can see that this is a minimal tree set because it contains all the splits. I mentioned K-compatibility, which some of you may be more familiar with, in which a set of splits is K-compatible if it does not contain a subset of K+1 pairwise incompatible splits. I just wanted to note that this is a different concept. So for instance, that set that we had earlier is 2 compatible because you can easily find 2 incompatible splits, say the first 2, but you can't find a set of 3, whereas it has a minimal tree set size of 3. So these are 2 different ideas of compatibility or incompatibility. And you can see it's not hard to show, for instance, that the minimal tree set size has to be larger than the K in the K compatibility. But I would argue that the minimal tree set size is a bit more natural, more sellable to biologists, I guess. So this, none of that actually answers the question that the biologist asked me. But the general idea here I wanted to know is, how bad can the incompatibility get? If your set of splits is the set of all splits on your taxa, how many trees does it take to represent all of that? And then we can use that as a sort of normalizing factor. And that way we can compare the incompatibility of two sets of splits corresponding to splits tree networks that potentially have different numbers of leaves. So this would be a very short talk. Oh, just quickly, sorry. This is a minimal universal tree set on 5 leaves. So it displays every single split on 5 leaves there. You can pick whichever one's your favorite and find it somewhere there. So to summarize, the question I wanted to answer was, how do we find the size of minimal universal tree set for a given set of taxa of size n? This would be a very short talk if there wasn't an answer. So here's the answer, and it turns out to be quite nice and short function. And so for the remainder of the talk, I'm just going to go through a toy example to give you the idea of how we proved it. It requires a short journey into POSETs. Sorry?
Just keep going, Mike.
Okay. Requires a short journey into POSETs. I'm going to assume that you're familiar with POSETs, but maybe not so much with chains and anti-chains. So a chain is a subset of a POSET where every pair of elements is comparable, and an anti-chain is a subset for which no pair of elements is comparable. So here is a nice POSET diagram, the Hasse diagram of the power set on 4 elements, where there's an edge between 2 things going upwards if the lower one is included in the upper one, for instance, 1 and 1, 4 here. So a chain is just any path, this is the set of elements contained in any path going vertically, say empty set 1, 1, 4, 1, 2, 4, 1, 2, 3, 4. And an anti-chain is a set of elements that there is no chain between any pair there. So for instance, an anti-chain might include, an example of an anti-chain is this row in the middle here with the 6 elements. I will shortly explain why we're talking about POSETs instead of splits. But before that, I just wanted to make 2 quick comments about anti-chains, which are going to be relevant. The first is due to a theorem of Sperner from 1928, that when you are looking at the power set on a set of size n, then this has size n choose n over 2 floored here, which should look familiar because it's in fact exactly twice the function we saw earlier. And secondly, there's a theorem of Dilworth from 1950, which states that if you have a POSET with the largest anti-chain having size r, then it can be partitioned into R chains. So taken together, what this means for a power set is that your power set can be partitioned into this many chains, n choose n over 2. So if we take that power set that we looked at earlier, we can partition it into chains like these. Here's one chain, here's another chain. These 2 over here are just chains consisting of 1 element. And here they are all nicely set out. And so the reason...
Michael, you've moved to...
Shit, no, I did it.
Are you there, Michael?
Yes, I'm still here. I didn't say anything interesting, so that's while I was muted. Okay, so yes, the reason we're looking at the Hasse diagram and the power set is because there's a supernatural way of associating each of these sets with a bipartition, which is say take 1, 2 here, we associate that with the bipartition where one side is the elements in the set and the other side is the elements not in the set. So 1, 2 gets mapped to 1, 2, 3, 4, as you can see here. However, as you can potentially also see, there's a lot of doubles in here. We have 1, 2, 3 versus 4 up here and 4 versus 1, 2, 3 down here. And that's happening there just because the set down here, 4, is precisely the set of elements that are not contained in the set up here, 1, 2, 3. What we're going to do is get rid of the splits that are above the central line here, and we're left with this. So why have we done that? There is a nice lemma, which I'm going to state here, which is all splits in one of these components that we're going to show very shortly can be displayed by a single tree. The only issue here is that, again, we still have some pairs of repeated elements. You've got the 1, 2 versus 3, 4 over here, and 3, 4 versus 1, 2, which represents the same bipartition. So once we match them up, then we're in a position where we can apply that lemma, which is that any split in this component is going to be compatible with each of the other splits and can therefore be represented on the same tree. With that, that's the part that requires a little bit of work, especially in the odd case where you have to appeal to some theorems on matchings. But for more information on that, you can check out the paper, which I'll link to at the end. So to summarize, we get this nice formula because we can associate a tree to special pairs of elements in the largest anti-chain of the power set on n elements, which is a bit of a mouthful, but I think is a quite nice and cute result. So jumping back to the original question on normalization, now that we know how bad it can be, what's the worst possible number of trees you need to represent all splits, we can use that to normalize. So for instance, this is a splits tree diagram on a set of archaeal genomes. It has 13 taxa. This is the same one, except we've just removed the bottom one down here, the Myrthanacoccus maripaludis. So it's, as we've only removed one, obviously got 12. And this first one requires 4 trees to display, and the second one requires 3 trees to display. But the difficulty is how can you compare them when they've got different numbers of taxas? But now we can. We just plug 13 and 12 into the formula. We can see immediately that the normalized value for N1 is less than the normalized value for N2. And so from this perspective, which is admittedly not necessarily a complete perspective, but is at least some first steps, N2 is more incompatible than N1. You can find the paper available on the archive now, and thank you for listening. It looks like there were some questions in the chat.
Thanks. There's some other comments in the chat. Thanks for a great talk, Mark. It's an amazing example, I think, of like a simple idea where there isn't an existing formula, and Phylogenetics throwing up just a really nice problem in combinatorics.
Yeah, it is the sequence is online on the OEIS, but there's no mention at all of anything to do with trees.
Right.
Yeah. So I was pretty happy with it as well.
Yeah. Are there any questions from the audience?
I have a live one. Go with it.
Barbara has a live one.
I'm just wondering how do you compute the minimal number of trees? That seems like an interesting, like the kappa function.
Yes, the kappa function. So unfortunately, I don't have a particularly nice way of doing that, and that's one of the things that I would like to do. For these particular cases, there weren't too many splits, and so I just went through them by hand and worked it out. I did. Yeah,
so another interesting problem in its own right. Yeah.
Yes, cool. I think there's a few natural sort of questions that come out of this.
Thanks, Michael. I think that's great. Yep. So we'll leave it there. And thank Michael again.
Thank you.
So we go from one Michael on one side of the world to another Mike on this side of the world with Mike Charleston, who I think perhaps needs little introduction, but Mike Charleston of the University of Tasmania. He's going to be talking about doing some gardening in split space.
Yes.
And I'll just say, with the speakers, I'm sending the speakers a message just to give you the 5-minute warnings. So if you're speaking, then just try to keep an eye on the chat box if that's possible. But otherwise, we'll just sing out.
Make that go on there?
So I know it can be difficult. I'm having trouble myself keeping track of everything. Yeah. And just a reminder to turn your mic off while the talks are on.
Yeah, there we go. Cool.
Okay, so Mike Charleston.
Right, cool. And where's the buttons back and forth?
Yes.
Cool. All right.
And you're on the screen? Yes.
And I'm on the screen. Fabulous. Right. Hi, everyone. Lovely to be here. That's because in Hobart, and it's lovely to see everybody's faces again, some familiar faces. I'm going to talk about landscapes in split space, not actually very much gardening. I'm just a little bit obsessed with my garden at the moment. But anyway, so splits we now know about, which is great, and we know about a little bit about trees. And so this stems from sort of conversations in previous Phylomania conferences, I think, about finding particular splits. And because I like to keep things, nice, simple problems. It's very hard to find one of some large number of trees, but it could be very easy to find a decent looking split. And if we can find good looking splits, branches that are in the right tree, and then subdivide our tree problem, I can maybe use the sort of divide and conquer approach to doing phylogenetics, like we can with sorting. Sorting goes from n squared for stupid sorting methods down to n log n for quick sort and merge sort. So divide and conquer might be great. Let's have a look. I think that's pretty much said. I've been thinking about landscapes of problems for a very long time, and so we're going to look at the landscape of this sort of way of thinking. So I think I don't need to say what a split is, but I've got a slide, so I may as well say something about it. For here, at least, I'm going to be talking about splits that are non-trivial, in a sense I'll describe, bipartitions of my taxon set. And the non-trivial part means it's a little bit of a fudge. It just means that each side of the split has at least 2 elements. So I'm not talking about leaf branches or pendant branches of a tree. I'm talking about inner branches of a tree. And I'm only talking about unrooted, fully resolved binary trees for now. And I guess what's important to note is that while the number of trees is enormous, as we know, it grows in a double factorial, the number of splits doesn't grow as badly. It's still enormous, but it's not as enormous. So it might be easier to search. And I'm just going to make a note that I don't really care. I want to emphasize, I don't really care what else is going on in the tree. I'm just looking for one split at a time. So in order to look for a good split, I need some kind of score function. I'm going to be talking about 3 score functions that I've thought about and coded up. So now I can think about this landscape in sort of complete terms. So the landscape is a graph, and the vertices of that graph are splits, and the edges between vertices are going to be determined by whether I can convert one split into another split by some kind of perturbation. All right, so very straightforward. And these perturbations are reversible in the sense that if I can turn one split into another, then I can turn the other into this one again. So the graph is an undirected graph, and it's going to be vertex weighted by the scores of each split. Okay. Now I could search through a landscape rather like hunting for the best phylogenetic tree, but by looking at neighbors and seeing if I can get to an improving neighbor. And I keep doing that until I can't get an improvement anymore. And then I've reached a local optimum. And so I would like to know, thinking about the landscape, how easy is it to find a local optimum? How easy and how many local optima are there? I mean, is it a nice, smooth, well-behaved landscape, or is it rubbish and I'll never find good splits looking in this way? So that's where I'm aiming with this sort of study. And I want to talk also about the last concept, really, which is the domain of attraction of a solution. So in this case, I'm talking about splits, and the domain of attraction for a locally optimal split will be all of the splits from which, if I do a steepest climb or steepest descent, I get to that locally optimal split. So everything in that domain of attraction is going to be of interest to me if it can get me to that locally optimal split, because I'm still hoping that that locally optimal split will be a good one. How am I going to be perturbing splits? Well, there's a few different ways I could do it. I'm aware that there's a big effect of the size of the split, which is the size of the smallest half, if you like, of the number of elements in the smaller half of the bipartition. That size has a big effect on the score for all of the split scores that I'm using. So I don't want a perturbation that's going to change the size. It just keeps it as it is. And the perturbation that I'm choosing here is just to flip two elements from one side to the other, just swap them over, as in the little figure here. So I've got a 3 and a 4 swapping. I don't know if that cursor is going to work. So all the splits in a given domain of attraction will be the same size. Here's a little example, a toy example. Well, it's a real one from the experiments, but it's very tiny. So I've got my correct tree on the left. I've shown, I think you can see the leaf labels, 0 to 5. I've labeled, I haven't labeled the internal branches, internal nodes, sorry. I've got the branch lengths given there, 0.05, 0.02, that kind of stuff. And on the right, I've got my landscape. And so, well, it's not the whole landscape, it's just the climbs. If I were to show all of the edges between all of the possible splits, it gets too dense and it's really hard to see what's going on. So here, an edge exists if a steepest descent goes from one vertex to its other end on the other end of the edge. Right, so this is only the climbs. So let's have a look at in a bit of detail about it. I've got a key on the right, which you can largely ignore, except for the coloring. And the coloring, which is it looks like a rotated 2 for reasons. There's a sort of a grey, greeny yellow color. That's a minimum score. The greeny yellow is the best score. And up to this rather violent pink color, that's the worst possible score. And all the graphs that we'll see in this talk are color-coded the same way on that 0 to 1 sort of scale. It's just normalized for comparison. Okay, each blob, because of the way this is set up, is a domain of attraction. So I've got 4 blobs there. I've got 3 internal branches of the tree. I'd like it if they matched up perfectly. They don't quite. So the first one to talk about is split 013 is not a branch of the tree, but it is locally optimal. If I searched, I might find that one, and I might conclude that that was a split in the true tree. But the other 3 blobs, the other 3 domains of attraction, all correspond to branches in the tree. So split 01 is correct, and it's easily found, and it actually has the best score under this scheme. 2-3 is a branch of the tree, and it, much the same way as Michael's talk, I'm just defining the split by 1/2 of it, so I can do that. It's in the tree, but it's a tiny domain of attraction. I'd be lucky to find it. And the last one, the 4-5 split, also in the tree, and moderate chance of finding it. So I really want to know, how often can I find locally optimal splits that correspond to branches in the true tree, and how often are branches in the true tree locally optimal? And how are all of these things affected by n, the number of species, the sequence length, maybe the model, branch lengths, and so on. So I only have a time to talk about a couple of these things. A little bit on the experimental setup. Just taking note of my time, good. So I'm basically growing trees with one program, growing trees under a Yule birth-only model, molecular clock assumption, different heights, different number of leaves, and then I'm generating sequence data under a really simple Jukes-Cantor model on those trees. And for each combination, I generate a bunch of different alignments, and then I feed those into the landscape analysis sort of program, which I've spoken about before. It parses the FASTA output and does this search. And it does a little bit of shortcutting by starting at each possible split and does a steepest descent, steepest climb, until either it gets to a local optimum or it reaches the split that it's seen before, and then we just don't need to do anything because we know what's going to happen next. We just make a note and store all the answers output in CSV files for throwing into R. My 3 score functions, two of them are really simple, one of them is terribly clever. One of them is frequency based. It's forming a vector of the base frequencies of characters, states, A, C, Gs, and Ts presented on either side of the split, and wanting to maximize that difference. The second one is essentially a parsimony-based method, and it's for a given pattern or site pattern, work out what is the best possible parsimony score for any tree that has that pattern and that split, and sums over all of the patterns. So there's a frequency-based and parsimony-based. And the third one is a SVD, singular value decomposition, a method based on Jeremy's subflattening approach to finding which splits actually might or might not correspond to branches of the right tree. 5 minutes, awesome. So those are my 3 methods, and I set up SVD first, and the name Flatbush of the program comes from the subflattening. So, in case you were wondering, so I did a bunch of simulations, and through the power of Bash, and able to get these running on the server somewhere, and then I was able to download my results for fairly moderate size of problems. N is 4 to 10. The height of, length is next, sequence length, 100, 320, 1,000, up to 32,000 bases. A height from 0 up to, from 0.1 up to 1, just to give a range of potential branch lengths. 10 simulations done on each of those combinations, and then applied each of the 3 different split scores, so sort of 7,500 odd simulations. I'm not going to show you them all. I'm going to show you sort of a collated summary of some of the output of that. So this plot is showing for the 3 different methods and broken up by the 6 different sequence lengths, roughly what proportion of true splits, the splits that are in the tree, turn out to be local optimum. So it's answering one of those questions. And so we can see, I'm trying to throw, this is my first time using ggplot, so it was steep learning curve. So we've got the diamonds show the mean proportion, and this is the proportion out of all possible true splits for the trees, for n is 4 up to 10 inclusive, plus some jitter to make things a bit easier to see. So the pink one is the frequency based, the green one is parsimony based. Parsimony ends up doing the best because in this sense, and SVD was disappointing because although they all increased, it wasn't actually quite as good as the naive methods. It just goes to show. Never can tell with these. Right. So the other nice thing about this is it can produce some lovely looking figures. This is one instance of the output for each of those sequence lengths, one particular height, one particular run, and the 3 different score functions. So I'm just going to pull out the last column of this, just for the L equals 32,000, just because I may as well pick something, and we can just see what we can learn from that. So the frequency one on the left and parsimony both seem to be relatively homogeneous in their scores for a given split size. So there's clearly a big effect on the split size to that score. Interestingly, the SVD seems to be much less affected by that split size, so and it's much more heterogeneous. You can see there's more colours within a single blob. So, interesting. And frequency tends to give generally higher or better scores for most splits, and parsimony for fewer. Haven't had a chance to explore that fully yet. We do have a few things we can read though from that. So first of all, clearly all of the methods improve with improved sequence length and branch length. That's nice. The parsimony and the frequency base both look promising. Power is way fast, which is kind of cool. There's obviously that size bias. And one nice thing that you might not have realized is that if I look at those blobs, those domains of attraction, the distance from any particular point to the middle, the locally optimal split, isn't very far. It's like 3 steps or 4 steps at most in there, which is kind of cool. It means that we might be able to find those locally optimal splits very quickly. So it's really interesting, and I think it's worth pursuing. And that's me done.
So thanks very much, Mike. You might like to
Do what?
Where are we? Oh, yeah, here
I
we
don't
go.
know.
Sorry, no, sorry. Great talk. Thanks for trashing my method. No, just joking. So,
Didn't tell it.
no questions for Mike. Okay. No, has anyone got a question for Mike? Just a very quick one. We'll get Christina. Barbara again.
So you're going to end up with a bunch of, I'm the best split in my domain of attraction. So you have a best 3-3 split and a best 2-4 split or so on. But you're still eventually going to have to face the problem of putting them back together if you want a tree.
True.
Any thoughts on how to, and in a sense then is the fact that the SVD one is actually not so biased by split size, maybe a point in its favour at that point?
Well, it might be. Actually, I'm thinking that I might aim for balanced splits to start with, because then if you can chunk up your problem into things that are roughly half size, then it's more efficient to then join them up. So I'll actually be focusing on one particular split size, probably. That's a good question.
Elizabeth Altman's jumped in with what I'm going to take as a comment, which was that she might find SVD work better if you had a more complicated sequence model.
That's
like
true. That's very
GTR,
true. It might.
markers.
Yes. Good point. Thank you.
Okay, so but we'll just save any further questions. Sorry, Laura, for later. I'll just get Christina up now,
I need to stop sharing.
and if Mike stops sharing,
Do it. I'll just get out of the way.
so Christine, you could maybe just say hello for the moment.
Yeah. Hi, I'm Christina. So I guess some of you know me. So I'm currently in Columbus, Ohio, where I moved a couple of weeks ago. And before that, I was at the University of Greifswald and did my PhD. And now I'm a postdoc.
Thanks, Christine. So we're actually gone to the other side of the world now. So
Yeah.
we've been to Germany, now back to Hobart, and then back over to the US. So I think you're ready to go, Christina. So it's all yours.
Okay. So you can see my screen?
Yep.
Okay, thanks. Yeah, okay. So I'm going to talk about some recent work with Arne Moors and Mike Steele on measures of biodiversity, namely feature diversity and phylogenetic diversity. And so one of the reasons why people are interested in studying biodiversity is really for conservation efforts. So given limited financial means, one central aim in biodiversity conservation is to conserve as much biodiversity as possible. However, it turns out that biodiversity in itself is not a very well-defined concept, and in particular, it's not easy to measure. And so in order to conserve as much of it as possible, we both need a way to measure or quantify biodiversity, as well as to assess the importance of certain species to overall diversity. And so there's a huge literature on different concepts. There's species richness, species abundance, phylogenetic diversity, feature diversity, trait diversity, functional diversity, and so forth. And so today I'm going to talk about two of those concepts, namely feature diversity and phylogenetic diversity, and how they relate to each other. So what is feature diversity? Well, assume that we are given some finite set X, of taxa, and additionally assume that each taxon has an associated set of fx of features. So this could, for example, be certain morphological or genetic traits. And we also assume that each feature has a strictly positive score assigned to it, reflecting the novelty or complexity or simply importance of a certain feature. And then we can summarize the sets of features across all our taxa by basically considering an n-tuple of those feature sets, and we call that a feature assignment. And such a feature assignment basically conveys the same information as a simple presence-absence matrix. So in this example, we have 3 species. Species A has features F1, F2, F4. Species B has features F1 and F3, and so forth. And now, given this setting, feature diversity is a group-based measure that quantifies the feature diversity of a subset Y of taxa. So given a subset Y of taxa, the feature diversity of Y is simply defined as the sum of the mu values over all features that are present in at least one taxon in Y. And so assuming that a variety of features will probably, for example, make it likely for species to be able to adapt to environmental changes and so forth, it is a plausible idea, or it seems like a plausible idea, to try to conserve as much feature diversity or feature variety as possible. So maximizing feature diversity could be one aim of biodiversity conservation. However, there are certain challenges to that. So first of all, it's probably not clear immediately what or how to even define a feature, and in particular, which features should be taken into account. So which features are conservation relevant. So that's one side, that's a problem. And the other is a computational problem. So finding an optimal subset of size K, for example, that maximizes feature diversity is computationally hard. So that's related to the set cover problem. And so for that reason, in 1992, Daniel Faith introduced the measure of phylogenetic diversity as a sort of predictor for feature diversity. And in order to talk about phylogenetic diversity, we need the notion of phylogenetic trees. And here I consider rooted phylogenetic trees that are not necessarily binary. And what I assume is that all edges are assigned non-negative edge length, representing either time or some sort of evolutionary distance. And for technical reasons, we also assume that we have an additional root edge or stem edge. And now, given such an edge-weighted phylogenetic tree, the phylogenetic diversity of a subset Y of taxa is defined as the sum of branch length in the subtree of T connecting Y and, in this case, rho prime. So for example, if we have considered the diversity of the set consisting of elements A and C, they have a phylogenetic diversity value of 7. And so the idea is that sets of species that are somewhat spread across the tree will carry more diversity than closely related species. So how does this relate to feature diversity? Well, the idea is that if you try to maximize phylogenetic diversity, then you're likely to choose species that are spread across the tree. And then it might be likely that they also carry a wider variety of features compared to choosing species that are closely related and probably carry a similar set of features. So that's basically why Daniel Faith introduced phylogenetic diversity as a predictor for feature diversity. And phylogenetic diversity has the benefit that this problem I mentioned, finding an optimal subset of size K, can be easily solved for phylogenetic diversity. So here the greedy algorithm works, and that was independently shown by Mike Steele as well as Paddy and Goldman. Okay, so Daniel Faith then concluded in his paper that greater phylogenetic diversity will, on average, imply greater feature diversity. However, he also noted, and this is important, that this is not guaranteed to be the case. So he went on, however, this does mean that the diversity patterns for some features may be poorly predicted by phylogenetic diversity. And surprisingly, while this paper has been cited excessively in the literature, about more than 2,000 times, I think, the formal relationship between phylogenetic diversity and feature diversity had never been studied or formally studied. And so this is what we were interested in. And now recalling what I told you about feature diversity in itself is independent of a phylogeny or phylogenetic tree. But in order to link feature and phylogenetic diversity, we need some model of feature evolution. And so we will consider a very simple or simplistic model of feature evolution. So assume that F is the set of all features present amongst the taxa in X. Then we make two simplifying assumptions. So the first is we assume that each feature in F arises on exactly one edge of T. And the second assumption is that if a feature has arisen, it will never be lost. And we formalize this by introducing a map H that basically tells us which edge each given feature arises on. And now this is, as I said, it's of course a very simplistic model, but whenever a given feature assignment, so a given collection of feature sets for our species can be realized under this model, we denote it by writing that F equals F H. So it can be realized under this model. And it's important to note here that by it can be realized or it can be explained, I mean, I really mean it can be explained by that model, but not it has arisen under this model. So it could be that there are, for example, hidden losses and hidden regains, but we could still, and then of course it would not, the true evolution would not have followed that model, but it could still be explained by it. And it's easy to see that not every feature assignment can be realized on a tree. For example, the one I showed you on the first slides can't, but it's easy to characterize when it can under using some classical phylogenetic arguments. And moreover, this model allowed us to formalize the connection between feature diversity and phylogenetic diversity. So we could show the following theorem. We could show that a given feature assignment can be realized under this model for some function h, if and only if feature diversity is exactly equal to phylogenetic diversity for all subsets of species for a certain edge length assignment that assigns strictly positive length to all interior edges of T and non-negative length to all pendant edges and the stem edge. And so this really means that phylogenetic diversity will only capture feature diversity exactly or precisely if the underlying feature set can be explained by this model. And when this is the case, then H and L are both uniquely determined. So this theorem basically gives two reasons why empirically or in practice, feature diversity and phylogenetic diversity might disagree. So the first reason is that the underlying feature set or feature assignment simply cannot be explained by that model. And in this case, we can't make a phylogenetic diversity and feature diversity coincide exactly. So that's one reason. And even if it can, then the edge links still have to be carefully chosen. So if we don't choose them in that way, then phylogenetic diversity and feature diversity might again disagree. And of course, thinking about this is only, so now we've characterized kind of the formal connection between feature diversity and phylogenetic diversity, but this is only really a first step, because more important or more interesting now would certainly be to analyze to what extent phylogenetic diversity still captures feature diversity when the models of feature evolution are more realistic and more complex, or in an extremal sense, how much can feature diversity and phylogenetic diversity disagree, and which should you then base your conservation actions on? Okay, nevertheless, with that we could kind of formalize the connection between FD and PD, but as I said in the beginning, we're not only interested in measuring biodiversity, we're also interested in assessing the importance of certain species to diversity, for example, to rank them for prioritization. And this is what phylogenetic diversity indices. So a diversity index, either for FD or PD, is simply a non-negative score assigned to each taxon that sums to total FD or PD. So the idea is that a diversity index apportions the total diversity present among all taxa, and this can then serve as a prioritization criterion. And so we defined a very simple feature diversity index, phi f. And this is defined as follows. So phi f of x is simply given as the sum of the mu values of features present in x divided by the number of taxa that have feature f. So the idea is that the importance or novelty of a certain feature should contribute in equal parts to all species that have this feature. And this index is very easy to calculate. It might seem a bit arbitrary, but we could then prove that it corresponds to the so-called Shapley value for a certain cooperative game. Unfortunately, for time reasons, I can't go into the details about the Shapley value. But let's just say that the Shapley value is a well-studied concept in game theory, and it has certain nice properties and characteristics that kind of justify this feature diversity index. So that's for feature diversity. You can, of course, do the same thing for phylogenetic diversity. And in fact, phylogenetic diversity indices are very well studied in the literature. So there's also a Shapley value, there's an equal splits index, and then there's, for example, the fair proportion index. And I'm just going to mention the fair proportion index because this one is actually used in a real conservation scheme, namely in the Edge of Existence project established by the Zoological Society of London. And so the fair proportion index was defined by David Redding, and its idea is to kind of trace back the evolution of a species and then divide each edge length on the path from the root to the species by the number of descending leaves. So again, the idea is that kind of the information that is represented by a certain branch in the tree should contribute in equal parts to the diversity index of all species that have descended from that edge. In this case, for species 1A, we'd have 1/1 plus 1/2, and so forth. And again, the fair proportion index corresponds to a Shapley value, and this was shown by Fuchs and Yin in 2015, and was quite a surprising result back then, because up until then, it kind of the fair proportion index and the Shapley value were considered independent or different indices. Okay, so now we have a diversity index for feature diversity and one for phylogenetic diversity. So how do they relate to each other? Well, we have, the first thing we could show is that if our given feature assignment comes from a tree, so can be explained by a tree, then the simple feature diversity index is equal to the fair proportion index for the edge length assignment LH, and LH is the edge length assignment that was induced for the equivalence of FD and PD. The more interesting result maybe is, however, the following, that basically states that given a feature assignment F and any rooted phylogenetic tree T, we can always interpret the feature diversity index as a fair proportion index. So given that tree, that there exists an edge length assignment L such that the feature diversity index and the fair proportion index coincide for all taxa. And the important thing to note here is that this is even true if the feature assignment itself does not come from a tree at all, or cannot be explained by a tree at all. And just to show you an example, so this is the feature assignment I showed on the first slide. And you can easily check that it cannot be represented by a tree, but assuming unit value for all features, we get a feature diversity A of 2, for B of 1, and for C of 1. And now, no matter which of the 4 trees or 3 leaves we consider, we can always find edge length assignment, an edge length assignment such that the feature diversity A equals the fair proportion index of A, and so forth. And in fact, we cannot only find one such feature, such edge length assignment. In fact, we can find infinitely many ones. So that was a kind of, even though this result is based in the language of feature diversity and phylogenetic diversity, that was a nice mathematical result in itself. Okay, so with that, I'd like to thank you and like to thank Arne and Mike, and I'm happy to answer any questions.
Thanks very much, Christina, and sorry about my message to you that said 1 more minute, go. And I meant to say 1 minute to go. Are there any questions for Christina? I only have the very boring question, which is the sort of politically orientated one about, with this type of work, where one sometimes talks about a constraint where you have to make these measures of diversity simple, so they're easy to understand for the public to interpret and to get behind, to actually implement. Is there any of this sort of thinking in what you're doing here?
So as I said, for example, if the fair proportion index is used by that Edge project, I think one of the reasons is because it's simple, but at least they are now thinking of incorporating a more complex one that also takes into account extinction probabilities and complementarity between species. So I think there's a tendency to use simple indices, but yeah, at least like these initiatives are now also thinking of trying to do a little bit more complex things.
Okay, thanks, Christina. I think we'll thank Christina again and everyone else for the first session. And I think that's gone pretty well. So any comments about how it's going, feel free to put it into the chat. But we have a break now for half an hour.
Yeah, 25-minute break. So we return at 11 a.m. Tasmanian time. I guess you've all needed to convert already, but approximately 25 minutes from now, we will begin the next session. I will leave the Zoom session running. It's the same Zoom session for the same Zoom login for the next session. But in case people actually want to have informal conversations or whatever, I'll just leave this session running. We're probably going to grab a coffee and return. And I'll make sure I'm here 10 minutes before the session if the speakers want to test their screen sharing and so on. So thanks, and see you in 25 minutes.